Good morning, students. Now, today we'll start the light reaction of the photosynthesis. Now, before starting that, I just want to repeat a little bit back. So, this is the structure I have drawn. This is the chloroplast. This is the cell basically. In that, I have drawn this chloroplast. Only one chloroplast I have drawn. In that chloroplast, I have drawn this structure. These are the thylakoids, and this is the granum, and they are joined with the stroma. Now, I draw this one thylakoid in a detail beta. This is one of the thylakoid. I have just drawn this thylakoid membrane. It is double membrane. I have already discussed. And in the membrane, you will have these photosystems. Photosystem 2 and photosystem 1. I have already told you what is the meaning of photosystem 2 and 1. Then I have elaborated this one photosystem. All these pigments, chlorophyll B, xanthophyll, carotenoids, they are filled in this. Right? I have just elaborated this one photosystem. In one photosystem, you are having xanthophyll, chlorophyll, right, B, and all other pigments on both sides. In the center, it is called reaction center. And this is having two chlorophyll A molecule, which is very important, right? Now, these molecules are getting solar energy. They are trapping, clear? Getting that energy, electrons get excited, and those electrons are moving from one pigment to the another pigment, and finally, those electrons are get shifted to the chlorophyll A, right? Similarly from this side also, and energy of 680 nanometer that is absorbed by chlorophyll A. And when this chlorophyll molecules get energy, their electrons also get excited, and then those electrons are get just jumped from higher orbit, and they are collected by one special molecule is there, which is called primary acceptor, right? Those electrons are collected by that. Then that primary acceptor. Now I have just changed the diagram. So this is primary acceptor. I have just now have full detail. This is chlorophyll A molecule. Electrons get excited, and that is collected by the primary, you can say, acceptor. Now this primary acceptor, this electron is then just given to the, you can say, next plastoquinone. Clear? This electrons get plastoquinone, and then cytochrome, and then to the plastocyanin. Means electron is jumped from one molecule to the another. And you have seen that I have drawn it down. It means that regularly electron is losing energy. And finally that electron is given to the photosystem 1. Same chlorophyll A is there. So that electron is just entered in the, you can say, chlorophyll A molecule. Right? Now, the only difference is, now another thing which happens in this case, that now I just show you how these electron is moving. Firstly, primary acceptor that get the electron, it is also called pheophytin. Then that electron is given to the plastoquinone. Plastoquinone gives that electron to the cytochrome, and then to the plastocyanin. Now, this electron then moves to photosystem 1. Right? Clear up to this? Now, this plastoquinone, it will get H positive ions from the stroma. Stroma means outside the granum or thylakoid, the extra structure of the chloroplast. And then this hydrogen ion is given to the cytochrome. Clear? And then after that, this H positive ion, which is carried by the cytochrome, catched by it, and given back in the lumen. Lumen means this part, beta. It is just entered into that, because we are filling H positive ions in the lumen of the thylakoid. Clear? Then after that, you will see that these H positive ions are filled by the cytochrome into the lumen. Clear? Then, now on the other side, we know that electrons are given to the photosystem 1. Then chlorophyll A is accepting that electron. Clear? Now this electron then goes to same way as a primary acceptor, then it will go to the FD system. Clear? Then this electron is just, this is the wall of the thylakoid. Clear? Now at this wall you are having the NADP synthase enzyme. Right? This enzyme will help to convert NADP plus H to the NADPH, and this process occurring in the stroma outside your thylakoid. Clear? And inside the thylakoid, this electron is just moving to the FAD system, is just giving moving electrons to the NAD and converting it into the NADPH, which is used in the dark reaction. Clear, beta? Now, finally, this lumen is filling with the H positive ions as I have explained. Clear? So, H positive ions are regularly filled in this. Then, after that, what is happening? There is one system. Clear? F0 and F1 part. Basically, F1 is the ATPase enzyme. Now, when this lumen is filled with H positive ions, this is the only way from where H positive ions will come out. Now, when these H positive ions, because inside it is more, outside less, so pressure is created, you can say, or concentration gradient is there. Due to that, these H positive ions will flow very fast from this canal. जैसे डैम से बेटा पानी निकलता है और बिजली बनती है, and that energy is used by the ADP for joining the phosphate, and ADP plus phosphate, they are giving you ATP molecule. Clear? So this ATP and NADPH formed in this reaction, they are used in the dark reaction. Clear, beta? Thanks and have a nice day. I think you understand the light reaction. Clear? Next is dark reaction. I will show you in the next video. Calvin cycle. Clear, beta?